Welcome back, my dear students. Today we will explore the fascinating world of spicules in Porifera, the tiny skeletal elements that give sponges their shape, strength and protection. Using insights from zoological textbooks and microscopic studies, I will explain the types, composition, structure and functions of these remarkable crystalline structures or bodies. By the end of this class, you will understand how spicules not only support the sponge body but also play a key role in its classification and evolution within the animal kingdom. Spicules in sponge. The endoskeleton or internal skeleton of sponge is made up of tiny crystalline structures called spicules or sclerites. These spicules provide support and protection to the sponge body. Each spicule has an organic axis or central core around which inorganic materials like calcium carbonate or hydrated silica are deposited. They show great variation in shape, which is very useful in sponge classification. Types of spicules. Spicules are broadly divided into two kinds: megascleres, which are the large spicules forming the main supporting framework, and microscleres. They are smaller spicules scattered in the body wall or mesenchyme that don't form the main skeleton. Megascleres, which are the main skeleton element of a sponge. There are 5 main types based on their shape and structure. Monaxons, it have a single axis and grow in one or both directions. They may be straight or curved. Types: monactinal monaxon are called also known as style. They grows in one direction only. They are rounded at one end and pointed at the other. That is style. If the rounded end has a knob, it is called as stylostyle. If curved and thorny, it is known as acanthostyle. Diactinal monaxon are wrapped. It grows in both directions from a central point. In the pointed at both ends known as oxia. If it is lens shaped, it is known as tornate. It is rounded, it is called as strongyle. If it is knobbed at both ends, then it is known as stylote. Tetraxons are also known as tetractines or quadridites. It have 4 rays radiating from a common point, which are not in one single plane. All 4 rays are equal. They are called as calthrops. One long ray and 3 short ones called known as trine. If one small ray is lost, it is called as dyne. If long ray ends in disc, it is called as amphidisc. If long ray is lost, it is called as triactinal or triradiate spicule or triode. It is found in calcareous sponge. Triaxons are hexactinal spicules. It have 3 axes crossings at right angles forming 6 rays, which are maybe straight, branched or spiny. They are characteristic of class hexactinellida or glass sponge. Polyaxons, there are several equal rays radiate from a central point. Spheres are the rounded spicules with concentric growth around a center. These are the different kinds of spicules like style, monaxon, triaxon, hexactinal, tetraxon, amphidisc, etc. Desmus, they are special heavily silicified megascleres found in some sponges. They are formed by deposition of silica on a small basic spicule or crepice. Depending on the types of crepice, they can be monocrepid, tricrepid and tetracrepid. These are interconnected to form a network like skeleton called as lithistid. Microscleres, they are minute spicules scattered in the mesenchyme. They do not form the main framework, but they help strengthen tissues. Microscleres are mainly of 2 kinds: spires and asters. Spires are curved in one plane and they are spirally twisted. Common types include stigmas, which are C-shaped, toxas, which is bow-shaped, chelas with recurved hooks or plates at each end. If the chelas are of having similar ends, they are known as isochellas. If they have dissimilar ends, they are called as anisochellas. Sigma spires, they are spirally twisted sigmas. Asters. Asters are basically star-shaped spicules with rays radiating from a center. There are main 3 types: small centered forms, which include oxyasters having pointed rays, strongyle asters having rounded ends, and thylaster having knobbed rays. Large centered forms include spherasters, which have definite rays, and sterasters, which have rays reduced to small surface projections. Other forms include strepasters, which are short spiny monaxons, for example, spirasters, sandiasters, plesioasters, amphiasters, etc. Microrapts or microxias and microstrongyles are small diactinal types. Development of spicules. Spicules are secreted by special mesenchyme cells called scleroblasts. They may be of two kinds: primary spicules, which are secreted by a single scleroblast, and secondary spicules, which are formed by more than one scleroblast. Formation of monaxon spicules. In calcareous sponges, a binucleate scleroblast secretes the spicule. Calcium carbonate is deposited around an organic axial thread between the two nuclei. As the spicule grows, the cell divides into founder cell and thicker cell. Founder cells are located at the inner end, and it decides the length and shape of the spicule. Thickener cell lies at the outer end, and it deposits additional layers. After completion, both cells move away into the mesoglia. In siliceous sponges, spicules are usually secreted by a single cell called silicoblast. Formation of triaxon or triradiate spicules. They are formed by three scleroblasts that meet in a triangular arrangement. Each divides into a founder and thickener cells. Each pair secretes a small spicule, and all three unite to form a triradiate spicule. Formation of tetraxon and hexactinal spicule. In the tetraxons, a fourth ray is added to another scleroblast. In hexactinals, the hexactinal spicules form inside a multinucleate syncytial mass, which is derived from a single silicoblast. What is the taxonomic importance of spicules? Spicules play a key role in sponge classification. Based on their composition and structure, phylum Porifera can be divided into three main classes, such as class Calcarea. Spicules are made up of calcium carbonate. Class Hexactinellida, there are 6-thread siliceous spicules or hexasters. Class Demospongi, which have siliceous spicules and spongin fibers from the skeleton. Summary table. So Calcarea have triradiate or tetraxon spicules made up of calcium carbonate, such as Leucosolinia. Hexactinellid have hexactinal spicules which are made up of silica, example Euplectella. And Demospongi had monaxon or desmos type of spicules made up of silica and spongin. Examples include Spongilla and Euspongia. So my dear students, today we explored the fascinating world of spicules in Porifera, the microscopic skeletal elements that provide support, protection and structural integrity to sponges. We learned how these spicules vary in composition, shape and arrangement, and how they play a crucial role in the classification and identification of different sponge groups. If you enjoyed learning about this tiny yet significant structure, don't forget to like, share and subscribe for more such engaging lessons in zoology. See you in the next class, and as always, keep exploring and stay curious. Thank you.